Genetically speaking, you are almost identical to a mouse. I mean, you share the exact same genetic toolkit.
Yeah, you really do.
But, one of you is listening to a discussion about cutting-edge neuroscience right now, and the other is, well, currently trying to navigate a cardboard maze for a piece of cheese.
Right. Slightly different Tuesday afternoons.
Exactly. Welcome to the Deep Dive. Today, we are looking at a massive, incredibly fundamental question. If the parts list is basically identical, what exactly makes the human brain so uniquely human?
It really is one of the most profound mysteries in biology because for a long time, the underlying assumption was just that to build a radically more complex brain, you would logically need, radically different genes.
Right, like entirely new blueprints.
Exactly. New blueprints that somehow uniquely coded for advanced cognition.
Yeah, you would assume we possess some proprietary, rocket science gene that mice simply missed out on during mammalian evolution. But the source material we are examining today, it just completely dismantles that assumption.
It really does.
We are unpacking a 2026 paper published in Nature by Javed and colleagues, and it's titled Developmental Gene Expression Patterns Driving Species-Specific Cortical Features.
A bit of a mouthful, I know.
Definitely. But our central mission today is to explore their core premise. What if human uniqueness doesn't come from having new genetic instruments, but simply from playing the exact same instruments at a different tempo?
Which represents a fundamental shift in how we view evolution. I mean, instead of endlessly searching for what novel genes we might have, this research pivots to looking at when and where we deploy the genes we already share with nearly every other mammal on Earth.
So to figure out what evolution actually tweaked to make our brains distinct, the researchers first realized they needed a map.
Right, but not just a static map.
Exactly, because building a brain is a dynamic, moving target. So they set out to map a developing mouse brain against a developing human brain, comparing apples to apples.
And that is an incredibly daunting logistical challenge. You can't just lay a fetal mouse brain next to a fetal human brain and, compare them day by day.
Because the timelines are wildly different, right?
Exactly. Mice mature at a blistering pace. Their entire corticogenesis, so the intricate process of building the neocortex, the brain's outer layer, that happens in a matter of days.
Wow.
Yeah, but in humans, that exact same process takes months.
So to look under the hood, the researchers utilized single-cell RNA sequencing. And for those of you listening who might only be familiar with traditional sequencing, think of old-school bulk RNA sequencing, like taking a complex fruit smoothie and trying to guess the recipe just from the taste.
Which is pretty much impossible to do accurately.
Right. Single-cell RNA sequencing is like sorting the fruit out before blending it. You can see exactly what each individual cell is doing at any given millisecond.
It's revolutionary. They analyze the active transcripts in 30,000 mouse cells from embryonic days 12 to 17, and about 28,000 human cells from post-conception weeks 12 to 24.
That is a lot of data.
It is. They also integrated data from lab-grown human cortical organoids.
Those are the 3D stem cell-derived models of developing brain tissue, right?
Yep, just to have a controlled human environment to test their findings against. With all that granular data, they constructed a two-dimensional map, a cytotemporal map.
Cyto meaning cell, and temporal meaning time.
Exactly. They track the very specific lineage of cellular development. The starting point is the radioglia, or RG. These are your primary stem cells.
Okay, so if we use an analogy here for you listening, think of building a house. The radioglia, the RGs, they are your general contractors.
I like that. And then those cells transition into intermediate progenitors, or IPs.
So the IPs are the specialized subcontractors.
Right. And finally, they mature into glutamatergic neurons, which are the primary excitatory cells in the cortex.
The finished, furnished rooms of the house.
Beautifully put. And the researchers use very specific genetic markers to track this lineage. When a cell was expressing SOX2, they knew it was a radioglial stem cell, the general contractor.
And when it expressed EOMEs, it had become an intermediate progenitor.
Exactly. And when URD6 lit up, it was a finished neuron. But this brings us back to the timeline problem we mentioned earlier.
Right. How do you actually align a 5-day mouse process with a 12-week human process without just completely distorting the data?
That requires solving a problem evolutionary biologists call mosaic evolution.
Mosaic evolution. Yeah.
It's the reality that distinct cell types within a single organism can evolve and mature at completely divergent rates. A human liver cell might mature on a different timeline relative to a mouse than a human brain cell does.
So you can't just use chronological time. You can't just say mouse day 12 equals human week 15 because the developmental stages just don't line up linearly.
Precisely. You can't just stretch the mouse timeline on a rubber band to match the human one. The researchers actually solved this using a mathematical alignment technique called dynamic time warping.
Wait, dynamic time warping? That sounds like science fiction.
It does, right. But it's an algorithm originally developed for speech recognition. It was designed to match two people saying the exact same word at different speeds.
Oh, that makes so much sense.
Yeah. So instead of anchoring the data to a rigid clock, they anchored it to conserved physiological milestones.
Like checking off specific stages of a construction project regardless of how many days it took the crew to finish them.
Exactly that. For instance, in both species, the deep layer neurons of the cortex are born first, and the superficial layer neurons are born later. That always happens in that order.
So by using those physical developmental events as the anchor points, the dynamic time warping algorithm just stretches and compresses the spaces in between the milestones.
Yes. And that creates a unified framework where a specific mouse progenitor cell and a human progenitor cell can finally be compared side by side on the exact same temporal map.
Okay, so let's do a quick recap for you guys listening to make sure we're all on the same page. We have our map. We use single-cell sequencing to look at thousands of brain cells. We align the drastically different timelines using dynamic time warping. And we're tracking the journey from radial glia general contractors to intermediate progenitor subcontractors to final neuron rooms.
Perfect summary.
But building a map of roughly 60,000 cells across two species creates a totally new problem. No human can manually read that much biological data.
No, it would take lifetimes. You need a machine to spot the anomalies. So the researchers deployed a machine vision algorithm, specifically convolutional neural networks, or CNNs.
So they essentially treated the gene expression landscapes like pixels in an image, right? Just looking for gradients and clusters.
Exactly. The CNN categorized the genes based on their expression, variance, and entropy. It found that about 67% of the genetic landscape was non-patterned.
Just diffuse, patchy genetic noise.
Right. Another 3% was ubiquitous, meaning these genes were turned on everywhere, all the time, mostly handling basic cellular housekeeping like RNA processing.
Stuff every cell needs just to stay alive.
Yeah. But the algorithm isolated a crucial 30% of genes that were patterned.
Patterned, meaning they turn on at highly specific times and places to do specialized jobs like organizing synapses or directing a neuron where to migrate.
Yes. And this is where it gets crazy. When they analyzed just those patterned genes across the two species, the results completely upended traditional evolutionary assumptions.
How so?
They found that the early stage cells, the radial glia stem cells, are remarkably conserved between mice and humans. Evolution barely touched the foundational starting material.
Okay, so the general contractors are the same.
And the final products, the mature neurons, are also highly conserved across the species.
Wait, hold on. If evolution didn't touch the foundation and it didn't touch the final product, I would normally assume the resulting structure just stays the exact same size.
That is the logical leap, yeah.
If the start and end are conserved, how are we getting a massive human neocortex out of the exact same starting stem cells? It just doesn't add up.
It adds up when you look at the middle of the process. The algorithm revealed that evolution messed heavily with the middle managers.
The intermediate progenitors.
Exactly. The researchers found massive evolutionary divergence in this specific group. In fact, about half of the genes that are strictly restricted to the early radial glia stage in mice
have been co-opted and shifted forward into the human intermediate progenitors.
You got it. Giving those middle-tier cells a whole new set of tools.
Which creates an entirely new, expanded physical region in the developing human brain called the outer subventricular zone, or OSVZ, right?
Yes. You see genes like HOPX and FAM107A becoming highly active here, driving this massive expansion.
Let's ground this in a mechanism for the listener. Tweaking the intermediate progenitors is essentially evolution switching from simple interest to compound interest.
I love that analogy.
Right. If a primary stem cell just directly makes one neuron, your brain grows linearly. Simple interest. But if that stem cell instead makes an intermediate progenitor, and that progenitor can replicate itself a dozen times before each of those copies makes a dozen neurons, your brain growth suddenly goes exponential.
Compound interest. You get a massive neocortex without having to reinvent the underlying stem cell or the final neuron blueprint. It is an incredibly elegant evolutionary hack for exponential scaling.
It really is. Okay, so we've mapped the timeline. We found out evolution supercharged the middle managers, giving us exponential growth. But how? What is actually driving this?
Well, this broad strategy of shifting genes around to supercharge the middle managers is perfectly illustrated by one specific, highly potent gene they isolated in the dataset, a gene called GUNB.
GUNB. And GUNB is a transcription factor, meaning its entire job is to bind to DNA and control the expression of other genes, right?
Exactly. It's a regulatory boss, and it is a shared piece of genetic hardware. Both mice and humans possess it.
But its timing on that cytotemporal map is mutually exclusive between the species?
Yes. In the developing mouse brain, GUNB is expressed very late. It only turns on once the cells have matured into finished neurons.
Showing up in the third act of the play.
Right. But in the human brain, GUNB turns on incredibly early, right at the beginning of their radial glia progenitor stage, Act 1.
To test if this dramatic timing shift actually drives human brain development, their researchers couldn't just, observe it. They had to break it.
Science at its finest. They performed a cross-species experiment using electroporation.
Electroporation. So they literally delivered a tiny targeted electrical shock to fetal mouse cells to briefly open up their cellular pores.
Right, which allows them to slip human genetic instructions inside.
That's incredible. So they artificially forced JUNB to turn on early in the mouse radial glia, essentially forcing the mouse cells to mimic the human timing. What happened?
The results were immediate. By simply expressing GUNB early, it bidirectionally controlled human-like cortical features in the mouse brain.
Wow.
It altered the proliferation rate of the progenitors, and it fundamentally changed the timing of when neurons were produced, ultimately increasing the total neuronal output.
So just by turning on a pre-existing gene earlier in the timeline, the mouse tissue started acting human. But let's dig into the why for a second. Why does delaying neuronal maturation by turning on JUNB early actually matter for human intelligence? What does that delay physically do?
It matters because that delay changes the architectural makeup of the final brain. By slowing down the overall maturation process, the brain ends up increasing its specific output of late-born superficial layer neurons.
And those are fundamentally different from the deep layer neurons born early in the process.
Completely different. Deep layer neurons mostly project their axons subcortically.
Meaning they send signals down out of the brain.
Right, to the brainstem or spinal cord to execute basic motor commands. But superficial layer neurons primarily handle intracortical connections. They wire the cortex to itself.
Which is the physical basis for associative communication, complex thought, and integrating information across different brain regions.
Exactly. So this simple timing shift in JUNB profoundly impacts human cognitive capabilities. It prioritizes the mass production of neurons that talk to each other, rather than neurons that just talk to the body.
Okay, so we know evolution supercharged the intermediate progenitors to scale up the brain, and we know that shifting the timing of shared genes like JUNB creates the physical space for higher cognition. But this exposes a glaring mechanical mystery.
The hardware issue.
Yeah. The JUNB gene itself is physically similar in both species. The instruction manual is exactly the same. So what physical mechanism is telling the human JUNB to turn on early, while the mouse JUNB just stays dormant until the end of the process?
That is the million-dollar question.
Yeah.
The researchers immediately looked at the upstream regulation. Their first target was the promoter sequence.
You can think of a promoter as the physical landing pad on the DNA right next to the gene, right? It acts as the on-off switch.
Yes. And the most logical assumption was that the human landing pad had mutated to attract different signal.
That would be the simplest answer. The switch itself just got rewired during human evolution.
But that hypothesis completely failed. They took the human JUNB promoter sequence and inserted it into mouse cells. And it behaved exactly like the mouse promoter.
Wow.
Yeah, it stayed inactive in the early progenitor stages and only turned on late. The DNA sequence of the switch itself didn't hold the secret to the timing difference.
So if the switch is structurally identical, then the difference has to be the finger flipping the switch.
Exactly. They discovered an entirely different upstream regulator, a gene called IRF1.
IRF1.
Yes. This gene acts as the supervisor that flips the switch. IRF1 is present and highly active in human radial glia, but it is totally absent in the mouse radial glia stage.
So evolution didn't rewrite the JUNB instruction manual at all. It just hired a new supervisor, IRF1, to read it earlier in the developmental timeline.
And the researchers proved this with a stunning intervention. When they artificially introduced the human IRF1 supervisor into the mouse cells.
Let me guess, it immediately found the mouse JUNB promoter and activated it.
Right there in the early progenitor stage, exactly as it does in humans.
That is mind-blowing. The mouse genome already had the complete biological capacity to build a more complex human-like cortex. The hardware was sitting right there all along.
It just lacked the specific software command, the IRF1 supervisor, to execute it at the right time.
So for you listening, think about what that implies. This is a perfect demonstration of what developmental biologists call latent or poised programs.
Evolutionary advancements don't always require inventing complex new genes from scratch.
Yeah.
Often the potential is already poised in the genome of the ancestor, just waiting for a simple upstream regulatory shift to unlock an entirely new developmental trajectory.
Okay, let's do another quick recap. We've got our developmental timeline. We've seen how middle managers exponentially grow the brain. We saw that shifting the gene JUNB earlier gives us the neurons for complex thought, and the supervisor, IRF1, is what pulls that shift off.
Perfect.
But understanding this precise minute-by-minute cytotemporal map isn't just an exercise in evolutionary history. The paper details how this exact same map gives us an entirely new mechanistic lens for understanding debilitating brain diseases that affect millions of people today.
Yeah, and this is arguably the most urgent application of their research. The team took 922 specific genes that clinical research has solidly associated with 9 different human brain disorders.
922 genes.
Right. They took those disease genes and mapped them directly onto this new cytotemporal landscape. And they released all of this data publicly on a database called humus.org.
Humus, spelled H-U-M-O-U-S. Assuming that stands for human mouse organoid sequencing.
You've got it. And when they mapped these known disease genes onto the timeline, incredibly clear delineations appeared based strictly on when and where those genes are supposed to be expressed.
Can you give us an example?
Sure. For example, genes known to cause structural cortical malformations, like microcephaly, a condition where the brain doesn't grow to its full size. Those genes mapped almost exclusively to the intermediate progenitors.
Wait, which perfectly validates everything we just discussed about exponential scaling. If the intermediate progenitors are the compound interest mechanism that scales up brain size, and a mutation breaks a gene that operates inside those specific cells, you lose the compound interest. The brain physically cannot build enough mass, resulting in microcephaly.
Exactly. The mechanism perfectly matches the structural outcome. Conversely, genes tied to severe psychiatric disorders, like autism spectrum disorder and schizophrenia, as well as epilepsy, they didn't map to the early progenitors at all.
Oh, really? Where did they map?
They are heavily enriched much later in the timeline, specifically in the finished, mature neurons.
Meaning those psychiatric conditions aren't necessarily defects in the physical building or scaling of the brain's architecture. They're defects in how the finished neural circuits refine their connections and communicate with one another.
Exactly. And organizing diseases this way, by the temporal phase of the broken cell rather than just the name of the broken gene, it totally shifts the paradigm for clinical pharmacology.
Right, because if a disease originates from a gene acting in a fetal stem cell, trying to treat the mature adult neuron might be biochemically useless.
Exactly. And that precise logic leads to their most surprising and potentially controversial finding.
Okay, lay it on us.
When mapping genes heavily associated with Alzheimer's disease, most notably APOE and LMNA, they found robust expression not in the aging adult brain, but incredibly early in the fetal progenitors.
Wait, what? Alzheimer's is the textbook definition of a late-onset neurodegenerative disease. We conceptualize it entirely as a failure of old, aging neurons. But this data shows the genetic drivers of Alzheimer's are active when the human brain is just a few weeks old in the womb.
It does, and this forces the medical community to consider 2 profound possibilities. The first is that late-onset neurodegenerative diseases might actually have deep structural roots established during early fetal neurodevelopment.
That is wild. And the second possibility?
The second possibility is a biological concept called pleiotropy, meaning a single gene does one crucial constructive job during fetal development and then does an entirely different, potentially destructive job in the aging brain.
Oh, wow. So if APOE is pleiotropic, understanding its fetal role is absolutely critical, because attempting to simply shut off or alter an Alzheimer's gene in adults without understanding its foundational purpose could have massive unintended consequences.
Exactly. It's a phenomenal synthesis of evolutionary biology and medical genetics.
It really is. So to tie all of this together for everyone listening, we started with a fundamental question about human uniqueness. By using dynamic time warping to align fetal development across species, we bypassed the noise of chronological time.
Right.
We discovered that evolution didn't reinvent the stem cell foundation or the neuronal roof. It supercharged our middle manager, intermediate progenitor cells to exponentially scale up production.
Combat interest.
Exactly. We found that our massive cognitive leap comes from subtle timing shifts in shared genes, like the transcription factor JUNB firing early to delay maturation and produce more intracortical connections.
The cells that talk to each other.
Right. We isolated upstream regulators like IRF1 acting as the software unlocking latent hardware programs that mice actually already possess. And finally, by mapping all of this on humus.org, we can clearly see that the origin of brain diseases, even late-onset ones like Alzheimer's, is intrinsically tied to this delicate temporal fetal ballet.
It really reveals that the dimension of time is just as vital to our biology as the physical structure of our DNA.
Which leaves us with a critical, somewhat unsettling thought for you to consider. If our vast cognitive abilities and our vulnerabilities to devastating brain diseases are largely the result of incredibly delicate timing shifts during fetal development, what environmental factors, microplastics, or modern chemical stressors might be subtly altering those exact timings in developing brains right now?
It's a heavy question.
It really is. If a gene firing a few days early creates the human mind, what happens when modern environments disrupt that tempo? What does that mean for the ongoing evolution of the human brain? Think about that. Thank you for joining us on this deep dive. Keep questioning the world around you, and we'll catch you next time.